Hey everyone, and welcome. Today, we're diving into the incredible, and let's be honest, often frustrating world of proteins. They are these tiny little workhorses that literally build our bodies and power so much of modern medicine. But they come with a huge catch.
And that's really the central paradox, isn't it? How can something so vital, so powerful, and so essential for creating new therapies be so unbelievably difficult for scientists to actually handle? It's a puzzle that researchers are trying to solve every single day in the lab. So, let's talk about this protein paradox. You see, inside their natural home, the cell, proteins are these elegant, sophisticated molecular machines. They're perfectly folded, they're functional, everything's great. But the second you pull them out of that comfy environment, they become ridiculously fragile. They can fall apart, clump together into useless junk, and just completely lose their function.
And trust me, this is not just some niche academic problem. This matters a lot. Why? Because proteins make up more than 60% of all current drug targets. We're talking about everything from insulin to the most advanced cancer therapies. Our ability to create new medicines literally depends on how well we can handle these incredibly delicate molecules.
All right, so where do we even start? Let's follow a protein's journey, beginning with step 1, getting it out of the cell. Now, you should think of this like a high-stakes molecular heist. The mission is to grab your target protein without breaking it. For this heist, scientists basically have two main playbooks. First, there's the traditional cell-based method. You use whole living cells. This is awesome for getting really complex proteins folded just right, but man, it can be slow and sometimes you just don't get a lot of protein out of it. The other option is cell-free production. Here, you're just using the cell's protein-making guts. It's super fast, you have a ton of control, and you can make proteins that would actually be toxic to a living cell. So you can see each one has its pros and cons. Okay, heist complete. We've got our sample in a tube, but what did we actually get? This is where the detective work begins. We have to run an identity check to figure out what we have and, just as importantly, how good it is. And the very first question you have to ask is the most basic one. How much protein is actually in here? What's the concentration? I mean, you can't run any useful experiments down the line if you don't even know how much stuff you're starting with. This is where quantification assays come into play. For this job, there are two main workhorses in pretty much every lab, the BCA and Bradford assays. With the BCA assay, you're using copper ions that react with the protein to give you this really intense purple color. Its killer feature? It works even when you've got detergents in your sample, which is super common. Then you've got the Bradford assay. It's way faster and uses a dye called Kumasi blue. This one's your go-to if you have reducing agents in your mix. But, and this is a big but, it totally fails if you have those detergents that the BCA handles so well. So yeah, your choice really comes down to what else is swimming around in your sample. Okay, knowing the amount is a great start, but it's only half the story. The next really big question is, did we only get our protein of interest, or did we accidentally scoop up a bunch of other random cellular junk along with it? We have got to check for purity. And the absolute gold standard for doing that is a technique called SDS-PAG. I know, it's a mouthful, sodium dodecyl sulfate polyacrylamide gel electrophoresis. But the idea behind it is actually pretty simple. It's basically a molecular sieve that sorts proteins based on one thing and one thing only, their size. So how does it work? Well, it's a pretty neat four-step process. First, you coat all the proteins with a detergent, SDS. This unfolds them and gives every single one a uniform negative charge. Step 2, you apply an electric field. This pulls all those negatively charged proteins through a gel. The little guys zip right through, while the big, bulky ones get tangled up and move much more slowly. Third, you stain the gel with the dye that sticks to the proteins, making them show up as visible bands. And finally, you analyze the pattern. And this is the moment of truth. What you're hoping to see is one big, fat, prominent band, right at the size you'd expect for your protein. When you see that, you know you've nailed it. It's this beautiful, clear visual proof that your extraction and purification actually worked. Success. So our protein is pure, we know how much we've got, we should be celebrating, right? Well, not so fast. The journey is far from over. In fact, you could say the most dangerous part is just beginning. Now we have to play defense and protect our precious sample from an army of threats trying to destroy it. And here's the crucial thing to understand. Once a protein is outside that safe, cozy environment of the cell, it's just incredibly vulnerable. It can unfold itself, which we call denaturation. It can start sticking to its neighbors and form useless clumps. That's aggregation. Or It can literally get chopped into little pieces, which is degradation. And the worst part? All of this is permanent. There's no going back.
And the list of enemies is long. You've got proteases. These are like rogue enzymes with molecular scissors, just looking for proteins to chop up. Then there's oxidation, sudden changes in temperature or pH, contamination from tiny bits of metal. Even just shaking the tube too hard can physically rip a protein apart.
So to fight off all these enemies, scientists call in a team of molecular bodyguards. These are special additives that get mixed right into the protein solution. You've got stabilizers, like sugars, that help prop up the protein's structure, surfactants that act like a non-stick coating so proteins don't cling to the plastic tube, cryoprotectants like glycerol that are basically antifreeze for proteins, and other agents that shield against oxidation and grab any nasty metal ions. It's a whole protective detail.
And finally, with your bodyguards in place, you have to pick the right safe house, the right storage method. For just a couple of weeks, you might keep it in the fridge at 4 degrees Celsius. It's easy to get to, but it's risky. For longer, say up to a year, freezing it -20 with some of that cryoprotectant is pretty standard. For really long-term archival, you go to a deep freeze, -80. But you have to be careful. Freezing and thawing it over and over will wreck your protein. And the ultimate? Lyophilization, or freeze-drying. It can make a protein stable for years, even at room temperature. But it's pricey, and you guessed it, it doesn't work for every protein.
Okay, we have followed this entire painstaking journey from extraction and identification all the way to preservation. So now let's just take a step back and look at the bigger picture. Why does all of this meticulous work matter so much?
Because really, this isn't just about getting a pretty band on a gel in a lab. This whole process, this is the bedrock of biochemistry. It's how we figure out the fundamental rules of life itself. And it's how we develop, manufacture, and guarantee the quality of medicines that treat everything from diabetes to cancer. The art of handling these tiny molecules has a direct line to the safety and the power of the medicines we all rely on.
And that really leaves us with one final big thought. We're now entering this amazing era of super complex engineered proteins, things like bispecific antibodies and fusion proteins. They have unbelievable promise, but they're also more delicate and more complex than ever before. So the real question for the future is this. As our therapies get smarter and more powerful, how are we going to evolve the art of protecting them? The future of medicine might just hang on that answer. Thanks for joining me for this explainer.